Hi, Dr. Kiki. For my extra credit video, I'm going to be talking about the key differences between mitosis and meiosis. So in mitosis, it results in two diploid daughter cells that are genetically the same as the original cell that they came from, and it has the same number of chromosomes as the one that it came from. Whereas meiosis, it results in four haploid cells, so they each have half of the amount of chromosomes as the original cell did. And they can be genetically different because of crossing over. In mitosis, they only go through pro, meta, ana, and telophase once, so they only go through that stage once, but meiosis, they go through it twice, so they have pro, meta, ana, telophase 1, and then they have pro, meta, ana, telophase 2. Mitosis is the reproduction of somatic cells, so just your body cells, and meiosis is the reproduction of gametes, so sex cells, which meiosis is only used in sex cells, so it's only used with sexual reproduction, whereas mitosis can be involved in asexual reproduction because it is just the reproduction of somatic cells. So here's my little overview of mitosis drawing. You can see you start with one cell and then it ends up in two. So this is interphase. This is the cell just kind of chilling, growing. This picture shows prophase, which is where the spindle fibers are like beginning to form. Chromosomes are condensing. As you can see, they went from here to the little X shapes, and the nuclear envelope is breaking down. This is metaphase, which is where the chromosomes are just lining up in the middle of the cell connected to their spindle fibers. This is anaphase, which is where the microtubules are like kind of pulling the chromosomes towards the like poles of the cell. And then this is kind of telophase. I thought that this represented it okay, so I just didn't want to draw a whole nother one. It's just where the chromosomes loosen up and like a little bit of a cleavage furrow will start to form in animal cells, or a cell plate will form in plant cells, and then it goes through cytokinesis, which is the actual splitting of the cell into two. So the cytoplasm divides, and it splits the cell into two daughter cells. So meiosis, this is my kind of overview, and I did it from meiosis too. So here's the two cells that came from the one, and then here's the four resulting daughter cells. So there would originally be another cell up here that splits into two, and then these two split into four. So these are the phases. This is interphase. Once again, the cell is just growing. This is prophase, where the chromosomes are starting to condense and they pair up and they can cross over in this stage, which is what creates the genetic variation in the DNA and in the cells. This one is metaphase 1, which is just where the cells line up in the middle. This one is anaphase 1, which is where they're kind of pulling apart, and you can see the different colors. I colored the like certain little fingers of them to represent the crossing over. But the sister chromatids are going to stay together through this stage. And then telophase 1, which is where they're separating. And then cytokinesis, which is going to be where they fully separate. And then for meiosis 2, prophase and metaphase are going to look the same. Anaphase is going to look a little different because the sister chromatids are going to pull apart. So they're going to become separated into individual chromosomes, which is what's going to make it have half the number. And then that's really the only difference between meiosis 1 and meiosis 2. Other than that, everything is the same. So telophase and cytokinesis will do their second round. They'll end up with 4 daughter cells with half the number of chromosomes as your original cell.